Welcome, everybody, to our weekly webinar. So we're going to just switch it up a little bit today, and I'm going to give a few slide updates, and Sabrina's going to help moderate some of your questions. Just going to give it a minute to make sure we get all the participants on. We've had quite the last 2 days. I know several of you were part of this FDA meeting with patients and clinicians and companies. It was really good, a 2-day open discussion. So maybe if we have extra time, we can even talk about that a bit too, but all good. So we're going to talk about the platform trial, just a little update on where we are, and then take your questions. Just another huge thanks to everybody who's supporting us. We were really talking about collaboration, and this is like a role model project for collaboration. How many people are working together on this, and how many groups are helping to support it. And I hope this will be a model for solving some other ALS challenges and gaps of really a community coming together to move the field forward. So Molly, next slide, please. So I know we have kind of some standard updates, but today I want to spend a little bit more time on when do we get results from the trial, because we've had a lot of questions about that. So I'm going to spend a little bit of time on that and then fly through the other slides. So last week we had an update from Prolennia on their drug, and next week we have one on Clean, and we have scheduled for each of the drugs in the platform a chance for the people at the company to tell you about the science. I'll give you a little update on our sites and enrollment, and then how to reach out to us, and then we'll get your questions. Next slide, please. Actually, one back. Yeah, perfect. Yeah. So as you know, this is a platform trial where we have efficiencies because we share the data from the people on placebo between the regimens, so that there's fewer people on placebo, and we can easily add new regimens, so we don't have to start all from scratch. So I'll just go through this a little bit. So when someone comes into the platform trial, they're randomized to whatever number of drugs are open at that site. So right now, most of our sites have regimen A, B, and C available. Some of them now have D available, and they're all good drugs. We picked them from a global competition where we said any company or any academic with a drug ready for late phase testing, like phase 2, 3 in people with ALS, apply. And we were going to pick it on the best science, as well as really ensuring that the company or whoever was leading that knew the dose that would be most likely be effective in people. So it was pretty rigorous selection to get to these first 4. And anytime any other company adds, we have the same rigor, same kind of committee that reviews the science and the dosing and the target engagement, because we want every drug that joins the platform to be exciting and good science. So after someone is randomized to a regimen, you know which regimen you're going to be in, A, B, C, or D. And the names of the actual drugs are here too. And then there's a second randomization to active drug or placebo, and that's 3 to 1. And so that's much better than most trials, which can be 1 to 1 or 2 to 1. And that's for 6 months or 24 weeks. And then again, you'll know if you're on, let's say, the zilucoplan arm, but you won't know if you're on active drug or placebo. And you won't know until we know the results of the trial. And I wanted to talk about that because that has come up in these prior webinars. So let's say someone's in the trial and they're in the zilucoplan arm, the double-blind period. At the end of 6 months or 24 weeks, you have a choice if you want to go into open label extension. And what open label extension is an ability to get the drug for sure. So let's say you were in the 75% that was already on the drug, and you're just continuing it. If you were on the 25% in placebo, then you're going to be switched to the active drug. So everybody has active drug and open label extension, and that continues. We continue to give it to people if they want it until we know the results of the trial. If the trial is positive, then again, the goal would be to keep giving it to the participants while more analyses are done or FDA discussions happen. If it's negative, then obviously we would stop it at that point. Sometimes people, even in a negative trial, feel it might help, and then there's a discussion about expanded access for somebody. So we don't analyze the results for efficacy to know if it works or not until the last participant in an arm finishes their last visit. So right now, for example, we're still actively enrolling. You'll see there's over 200 people on drug right now, but we do need to get to 160 in each arm. And so when, let's say, the 160th person is in the zilucoplan arm, then we're going to have results, about 6 months after that, a little time for the analysis. But we won't know it when, let's say, the first person gets to the open label extension. We won't know whether the drug works or not. We will know, though, safety. So we have a data safety monitoring board that looks every 3 months at the data for safety. We've also built in something called futility looks, so that's an interim look that if the drug is going the wrong way, like if people might be doing worse on it, or if there's no chance of it succeeding, it would abort early. And I think every ALS trial should have that, and we insist on that. And so we have that for all of the regimens. I hope that's helpful, at least on when we'll know the results. So the faster the entire trial enrolls, the sooner we get the results. Now, I think if we weren't in the middle of pandemic, it would be even faster than it is right now. Enrollment's actually going very well, but there's obviously a lot of challenges because of the pandemic. Next slide, please. So we have one more site activated than last week, 38 sites. We have several more, about 5 or 6 that we're hoping will be activated next week or the week after. Again, we're trying to get all 54 sites up and running. And most of the issues for those sites that aren't activated are really related to the pandemic, about not having staffing or the ability to start in the pandemic. It's very, very different state by state, city by city. But everybody's trying. We are going to add more sites. We made a call for it, and we're going to be selecting more sites in the next month. It will take some time for the new sites to add, but we did want to keep getting more sites geographically spread out so that people don't have to travel far to be in the trial. If you want to know if there's a site near you, there's this website, and there's also our patient ambassadors who are on the call here that can answer any question and help you get connected to the center. Next slide, please. So these are the list of the sites. The spectrum, the one at the end, is the newest one. And again, we're trying to get even more activated, but 38 is a good number as well. Next slide. So we have 292 people who have signed consent. There's this kind of multi-step process. So of those 292 people, 234 have gone to been assigned to one of the regimens that are open, and 200 are already on drug. And we'll again keep you updated on this. Next slide, please. So I think our patient navigators. They're both on the line here, Catherine Small and Allison Boulat. They're available to answer any questions. There's a phone number and an email, and I know they've been helping many people find sites near you or find sites that have openings sooner to help and answer any questions about this trial. Next slide, please. As you know, the weekly webinar idea came from our patient advisory committee. We love doing this. We are trying to change it up a little bit, and we heard you also about having more information about the drugs that are in the platform trial. So last week we had our colleagues at Prolenia speak, and we did record that, and that's available on our website. Next week it will be the folks from Clean. They'll talk about their drug, and on the 18th, Biohaven, and the 25th, UCB Raw. They'll all be recorded and put on our website. Next slide, please. And we're open, now we'll open up for questions, but we're open for any other ideas on what would be useful to hear about at these brief weekly updates. So
Great.
Good. I'll go to Sabrina next.
Yeah, there's a few questions. The first question is, if a patient is taking AMX 35 and showing some improvement, can he be considered for other drug trials?
Oh, so the AMX, sorry, say it again.
If the patient is taking AMX 35 and showing some improvement, can he be considered for other drug trials?
Yeah. So AMX 035 is still considered an investigational drug in all countries. It's not an FDA approved drug yet. So right now it's exclusionary for every trial. If the FDA does decide to approve it, if the company applies first for approval and it is approved, then that will change, just like riluzole is allowed in any trial, and so is edaravone.
Great. There is another question about one of the components of AMX35, specifically bufenil, which is sodium phenylbutyrate. So the question says, in the bufenil pamphlet, it indicates that neurotoxicity is a possible precaution side effect. Wouldn't that be harmful to an ALS patient?
So in the trial that Sabrina led here with our team, we didn't see those type of side effects, but it was a small trial, and what's in the drug label is really from all their prior trials and other illnesses. It's a marketed drug for a pediatric disease, but in the trial we didn't in people with ALS. We did see a slowing of the illness, and the most common side effects were GI related.
If someone is getting Ibudilast from Japan, can they participate in the trial given that it's not approved here in the US?
No, they can't. So Budilast is an experimental treatment for in development for ALS. There's a phase 3 trial going on, and we don't have any data that it works. There's been some small studies by Dr. Brooks, and we did a small, actually expanded access program of it a couple of years ago. But right now, that we don't know that it works or doesn't work in people with ALS, so it is exclusionary. So the exclusionary criteria are on our website, but one thing I'll just say that the general principle for all trials is that if something's marketed and standard of care, of course people can be on it. But if something's in another trial, you can't kind of be in both, because in the end we wouldn't be able to tell if the new drug worked, and that would mean that we'd still be here 5 years from now not knowing if the drug worked or not. So that's the reason. But the good news is that trials are shorter now.
Yeah.
It'll be less exclusionary.
There's a few questions about the platform trial. So the first about the design, how is progress of the illness measured amongst the participants?
Yeah, good question. We have a couple ways to do that. One is with this functional rating scale called the ALS Functional Rating Scale. That's 12 questions that asks about different things that people can do. We're also measuring breathing function, and we have two ways of doing that, one in the clinic and then one at the home. And we're measuring strength. We're have some patient-reported outcomes about how people feel, and we also have a voice app. So one of the things we want to do in the platform trial, besides, the main goal is to find effective treatments, but we also want to keep learning about the illness and learning how to do trials even faster. So we've built in some of these new outcome measures to see if they might be able to shorten trials or lower the number of people needed to tell if a drug is effective or not.
Another question about the drugs in the platform trial. Do any of these drugs look like they're helping? And if so, how are they helping?
Yeah, so they're all picked on really good biology, okay? But this is the first clinical trial for them in people with ALS, with the exception of the clean drug, which is in a trial also in Australia. But we don't have any efficacy data on any of them. So meaning we don't know if they work or not. We know they have great science and rationale, but this is really the trial to tell if it works or not.
So about trial design again, what determined 160 as the magic number for each regimen? As new regimens are added, will directing enrollees to that arm of the study significantly delay the completion of the existing arms?
Yeah, good question. So we came up with the 160 by a couple things. One, we have 4 actually really brilliant statisticians we were working with, and we have the PROACT database, which is an open source database that Alex Sherman and our group created with Prize for Life, of all the placebo data from past trials. Why that's important is you can use that data to model the inclusion-exclusion criteria and what effect size you can see. And effect size is how much effect on the disease you can have. So we balanced this by saying we want to pick up drugs that slow this illness down by 30% or more. So we're not looking for 10% or 5%. We're looking for a pretty good size, it's 30% or more. And we wanted to be more inclusive because we heard from patients about the difficulty of being in trials that might have a 2-year cutoff or an 18-month cutoff. So with 160 people and the inclusion criteria we've picked, which is largely symptom onset for 3 years or less, and breathing 50% or more, we can pick up a 30% slowing or greater with 160 people per arm. The final statistics is actually 120 on drug compared to 120 on placebo, because again, we're pooling the placebo data from each of the regimens.
And again, about the, you just mentioned that, but there's a question about vital capacity. Does it have to be 50% for all of the arms of the trial?
Yeah, I mean, the beauty of the platform trial is that you can pool the placebo group, but to be able to do that, you have to have the same criteria for each of the arms. So it is 50% or greater for any of the regimens. Now I know. Sorry, there was a question about whether adding another regimen slowed down the recruitment for the current one. It does a little bit. It doesn't like hugely, and I don't think we're planning to have more than 4 enrolling at the same time. We might, we have a 5th one we're working on, but my hope is that the first 3 would be done enrolling before we added that. They might overlap a little bit with the 4th one and the 6th one.
And a quick question about enrollment. How many people are enrolled in each of the first 3 drugs at this time?
Yeah, it's about 67 or 68.
Right.
Yeah, so we still have a ways to go. I mean, it was enrolling really fast. I think the December and the pandemic getting worse has slowed it down a little bit, but it's still enrolling pretty well.
Yeah. There's a very sophisticated statistical question. On the counter side of a futility analysis, can you explain to people how the FDA's statistical penalty works for manufacturers who decide to do an interim analysis, and how you or a manufacturer decides if they will be doing those in the HEALEY trials?
Yeah, it's a really good question. I can explain it with hand waving, but not with the deep statistics. So if you look at the data 20 times, right? There's a 1 in 20 chance that you're going to find a positive result by chance. Okay, that's what the whole statistics, the p-value is. And so the more you look, the more chance there is that you're finding a spurious finding. So every time you look at the data for efficacy, the FDA requires that you take what's called a hit on your p-value, meaning that at the end of the trial, P of 0.05 or less is not enough. You have to be better than that. So companies don't like to do that because it makes it harder for them to show efficacy at the end of the trial if they look a lot along the way. And I'm sure a statistician can explain that much better than I do. So for a 6-month study, that's short. That's as short as we can probably do right now without better biomarkers. If you look after 3 months, the chance of your seeing an effect is very low because just not enough people have been treated for long enough. And then you also have to take a hit on your statistics at the end. No company, we offered that to all the companies, but no company accepted that. And I actually asked that to the FDA on our FDA conference, and they agreed that 6 months is too short a trial to take interim looks for efficacy. Now, it's a different story if you have a 12-month study. And a lot of companies do actually take an interim look when, let's say, half the people finish 6 months, they might take a look for efficacy. But it's a risk at the end of the study that you have to reach a higher bar if you look multiple times. Now, the flip of looking for futility or looking that it's going the wrong way doesn't cost you anything on your p-value. So that's why I think every trial should always have that.
Right. There's a couple of questions about biomarkers. I'll start with the first one. It's a little bit of a longer question. It says correctly that there have been many scientific publications on biomarkers recently. For example, there was one about the discovery of an MRI standard to determine the difference between ALS and other motor neuron diseases that mimic the same symptoms. And there's also research studies showing that TDP-43 can be measured in serum. What is your take on these and other biomarkers? Has there been any real success within this field?
Yeah, first of all, I think we desperately need them, and there's different types. There's diagnostic ones that might help you figure out if there's a certain group of people that a drug makes more sense for. For example, something like measuring HERV-K, which is a marker of a retrovirus. If somebody has high levels of that, maybe it makes sense for them to be in drugs that block that virus, that type of virus. So those are more diagnostic biomarkers. But then there's biomarkers of treatment effect. And that I think is we're inching towards some progress. And I think because so many patients have given their blood and spinal fluid, we have big repositories that we're giving out to people to try to find one. And the best one we have, I think, so far might be neurofilament. This is a measure of a protein that's in your motor axons and leaks out when they're damaged. And is high in people with ALS. And there's at least one study, a gene therapy trial, where it lowered that neurofilament level by 50% in the spinal fluid. And there, at least in that small study, looked like there was a clinical effect. Now we'll know for sure when that bigger phase 3 trial is done. And if it works clinically and also lowers neurofilament, then I think we might have our good biomarker. So
Yeah,
we're getting there. Yeah.
and there's actually a follow-up question on biomarkers. When do you consider removing the placebo and instead use neurofilament light or another biomarker instead of placebo?
Yeah. I can't wait for that day, I'll say, but we're not there yet. It has to be a really proven, what we call a surrogate marker, meaning that anytime you reduce that marker by X amount, it correlates with the clinical effect. And so I think if we had a drug that was very positive and clinically and lowered the neurofilament clearly, then that might meet the bar of a surrogate marker, but we don't have that yet. But it's included in every trial, including the platform trial. And we do ask people, if it's voluntary, to contribute spinal fluid in the platform trial just for this reason, so that we can measure neurofilament, measure other things. And then if and when 1, 2, 3, all 4 of the drugs, if they work, then we can compare it to that biomarker.
Yeah. If you want to give blood to assist in investigation of biomarkers, how is that done?
Well, first of all, thank you for doing that. So there are many centers throughout NEALS that have biomarker studies that are, working together to. So I guess one question would be finding out if your neurologist is part of that. If they're not, then I think I'll probably defer to Catherine. Maybe call Catherine, and we can try to connect you up with a center that is close. A lot of that can be done remotely, but some of it is more complex blood collection and might need to be done at the center.
If someone has familial ALS but is pre-symptomatic or asymptomatic, would it be beneficial to measure neurofilaments level, and how can it be measured easily?
I think it's a really important research study. I don't think that we know clinically what it means yet. So I just want to make sure people know that. But it's not like if you measure it and it's normal, it means that you're not going to get the illness. Or if you measure it and it's high, it doesn't necessarily mean that you're going to get the illness. So it's not a validated marker yet for risk of getting the disease. However, it's important for us to learn that. So I say there are some studies. It's called DIALS. DIALS, that is following people who are at risk for familial ALS, just to answer that kind of question.
There's a comment from a listener, a technical comment that I cannot hear. My volume is at 100% and I cannot hear. I can hear, but I don't know if there's maybe, Molly, if you can do a technical check, or we can all try to speak up, I guess. At least one listener cannot hear. There's a lot of great questions, so maybe we can, in the last few minutes, we can go over them. Somebody says, we've filled out all the paperwork to enroll in the trial. When we will hear that we are accepted into a screening?
Yeah, so hopefully you'll hear very quickly, but if you don't hear back from your site, please reach out to Catherine Small here or Allison Boulat, our patient navigators, and we'll help speed that up.
If a patient was in the trial taking either the drug or placebo, will they have the drug supplied to him or her at no cost after FDA approval?
Not after FDA approval, but let's say you do the trial and you're in the open label extension, which would be at no cost, and the drug is positive, then all our partners have said that they would continue giving it to people, and that's typically done from companies, an open label extension, or maybe switch it to expanded access until market approval. But once it's on the market and your physician can prescribe, those usually end.
Yeah, and on the technical note, I got some messages from people that can hear. Also, please note that the webinar will be recorded and posted on our website. So if you're having a hard time hearing, you can perhaps watch it later. We have seen the FDA approve COVID vaccines very quickly. What time frame can we expect to see on the drugs in the platform trial?
Well, the first step is we have to finish enrollment, and so that we can finish and get the results. But let's say we get the results and they're positive, then the companies. The next step would be the companies to talk to the FDA and submit an application. The fastest I've seen the FDA ever approve a drug from the day like a company submitted to when you could prescribe it was 9 months, and that was for a childhood illness called spinal muscular atrophy, another motor neuron disease. But during that 9 months, the company was providing it on compassionate use for people. So it can be fast.
Yeah, there's a couple of questions about placebo. So the first question is, since you have a shared placebo pool, isn't it possible to increase the active to placebo ratio as you add more regimens?
Yeah, that's a great question. That's our hope. I think we need to see in these first 4 or 5, we have to see that whether the placebo course changes or not. So what we, if it's stable over time, then yes. If it changes over time, then no, then we won't be able to.
There's a
That's a good question.
Yeah, there's a question about placebo and whether it's possible to completely remove it. And the question is whether ALS researchers are talking about the placebo research system. And the comment is this is a horrible system because ALS is only one direction and the placebo times are horrible.
Yeah, again, I think the platform trial is a huge step forward with the whole. It's the first one that at least pool placebos across trials and go to 3 to 1. The issue and why we don't have we can't get rid of placebos is because the disease is so different between people. We don't know enough about the biology to separate people out by biology, and drugs. None of the drugs yet, as far as we know, would like stop the illness. Like if you have a drug that stops the illness or reverses it, you don't need a placebo. You need one person. But we're not there yet. But everyone wants to get there. But to get there, we need more science. We need more understanding of the cause.
We have a few questions in the last few minutes. The first question is about the dose that participants will receive in the open label extension for the CLEANS regimen. So maybe I can quickly say it's either 30 milligrams or 60 milligrams. So both doses will be tested in the open label extension. And as a reminder, next week we will have CLEAN come and present on their drug.
Yeah.
Another question, is there anything that patients can do to convince their local trial site to participate in EAPS?
I think, yeah, you could, first of all, you should talk to them and let them know that you're interested in it. And then, as you know, we got a recent grant from IMALS to help teach sites how to do it and to how to template some of the work for them. It isn't nothing. I mean, it's important to do. We love doing it, but it is a little bit of work for the sites. So we're trying to lower that barrier. But I think hearing from their patients how important it is key. And also maybe your local ALSA or MDA chapter, because it does cost some money for the sites. And so maybe they can get some grants from the local ALSA center or MDA center.
How soon after the end of the 6-month trial will participants find out if they were on placebo or not?
Yeah, great question. So the FDA in particular asked us to keep people blinded to what they were on and to keep us blinded to what they were on in a double blind during the open label extension phase until we're done, until we know the results. And the reason for that is that in a way, it can be treated like a second study. And the Centaur or the Amelix study is a perfect example of that, so that you can learn so much more. In a way, it might help speed up getting approvals. So it could be a while. Again, the faster we enroll, the shorter that time is. So as soon as we know the results, basically.
There's a few questions about 2 other trials, not the platform trial. One is the Centaur Amelix trial, and the questions are whether Amelix will apply to the FDA for AMX 35 or if there is a phase 3 trial, and if so, if there are results of that. And the other one is about Neuron, any updates now that there is an EAP for that. I don't know if you want to kind of provide a general update about those studies.
Sure, I'll answer Neuro, and then you can obviously answer the Amelix. I'd say that I don't have any new updates. I mean, I'm very glad that they're providing expanded access to some of the people that were in the trial, but they're in discussions with the FDA, and there's no information that I know of how long that will take.
Yeah, same for Amelix and the AMX35. The company is definitely engaging in discussions with the FDA. And again, as those discussions happen, there's no real updates because all options are on the table, and we still need to see the results of those evolving conversations. But again, options could include, approval could include another trial or expanded access. We need to see the final outcome before we can really cannot guess the outcome. So hopefully over the next few weeks we will know more.
But we promised to arrange webinars with both those
Absolutely.
companies as soon as they have some information.
Yeah. The last couple of questions, I think, for today. If one or more of the treatments show significant improvement, how long before it's available to other people with ALS?
I think that will be an important discussion that we'll have with the companies. So let's say we have one of them is like a home run, and they're going to go and apply, which I hope all of them will be home runs. But let's say one of them is, and they're going to apply to the FDA for approval. Often companies will make it available on expanded access during that period of time because it can be a 9-month period of time, and we would certainly be advocating for that. And we're actually trying to set up the infrastructure to be able to do that for any drug that has positive phase 2, 3 results. Through NEALS and through the Healey Center.
Yeah, and I think the last question for tonight, for an EAP, after asking your neurologist to be entered, how long does it take? How long does this process take for them to set up an EAP?
I think on average it's 30 to 60 days. The next step would be, I mean, they can say yes, but they have to get permission from the company because the company owns the drug. So and then as soon as the company says yes, then they have to write a protocol, and that's what we're trying to template out for them in the Zion ALS grant based on the EAPs we have at the Healey Center. The FDA usually reviews within 30 days, and same for the IRB.
Great. So I think that was most of the questions. There's a few questions coming in last minute, maybe, quickly taking this. After getting permission, how long does it take for the protocol? Our EAP is taking months.
Again, that's what we're trying to do. Like we're all about speed and efficiencies and not having people duplicate efforts. So we have a template of an EAP that we're going to be sharing with our sites.
Yeah, there's also a question about the chemistry of phenylbutyrate and it's metabolized into phenyl acetate. Questions about why not using the metabolite or the prodrug. I mean, I would suggest for that, maybe you can also contact Amelix Pharmaceuticals directly. We are also doing some additional chemical analysis that we will certainly publish on the study. But I think if you have questions about the medicinal chemistry, you can also contact them.
Yeah, that one I definitely don't know the answer. But it's a good question.
Great.
Excellent.
Thank you.
Guys. We'll see you next week. Next week is, I think, Clean is presenting.
Yeah, great. Thank you.
Bye.
Bye.
Bye, everybody.